Hello again. Welcome to the third lecture for the week of the 31st of August. So I had two lectures on amino acids and proteins. This slide deck here is around 80 slides. It will contain both lectures for the enzymes, which I normally do over two lectures, but since we're going virtual here, I'm going to combine it into one. So this is following chapter 6 in your book. My email information is here if you guys need to get a hold of me. But again, we're talking about proteins, catalytic proteins, specifically enzymes today. Here's an outline of what I'm going to be going over in this lecture. First, we're going to talk about, at a high level, free energy, equilibrium, and kinetics, and how this applies to enzymes. The conditions that impact enzymatic reactions. We're going to go through the different types of enzymes, how enzymes are classified at a very high level. We're going to talk about the structure of enzymes, specifically cofactors, active sites, things like that. The properties of enzymes. We're going to spend some time on Michaelis-Menten equation. I'll tell you what that is and what the Michaelis-Menten constant is. It's a good way to describe enzyme reactions. We'll talk about the major types of enzyme inhibition. And then I'll end with some regulatory mechanisms affecting enzyme reactions, some allosteric effectors, covalent modifications of enzymes that can occur, and some other mechanisms of enzyme regulation. First, we'll talk about free energy, equilibrium, and kinetics. Enzymes are catalysts of biological reactions. They can accelerate reactions by millions of folds. So, for example, if we think about carbonic anhydrase, without the enzyme, the reaction without the enzyme, the rate constant is about 0.1 per second, so 0.1 turnover per second essentially. With the enzyme present, it's around 10 to the 6. So you see a very big rate enhancement with the enzyme present. Another example being chymotrypsin. We'll talk about chymotrypsin throughout this lecture, using it as an example along with trypsin. So without the enzyme, you see a very, very, very slow reaction. So this is a protease where this peptide substrate or the protein substrate gets cleaved. Without the enzyme present, that happens at a very, very slow rate. But with the enzyme, you get a much higher rate reaction. So there's obviously a huge acceleration in the rate. So that's basically what enzymes are for. It's to take a reaction that would be very, very slow without the enzyme present and to make it occur much more quickly. The common features for enzymes and inorganic catalysts include catalyzing only thermodynamically possible reactions. The enzyme or the inorganic catalyst that could be used like an enzyme, they are not changed during the reaction. They don't change the position of equilibrium and direction of the reaction. And they usually act by forming a transient complex with the reactant, thus stabilizing the transition state. And we'll talk about what that means. So at a very high level, if we think about the reaction coordinate of a reaction, a non-enzymatic reaction requires a lot of energy to complete that reaction. So the reaction coordinate just going from start to end, essentially. When an enzyme is present, that activation energy barrier is lowered, so that reaction can occur much faster. So that's a very brief introduction to free energy equilibrium kinetics. The conditions that impact enzymatic reactions, the information follows. The specific features of an enzyme, as we talked about, they accelerate reactions much higher than if you don't have any enzyme around. And even in the presence of some sort of inorganic catalyst, enzymes are much faster at accelerating reactions. They're very specific in nature, so they're very specific to the substrate or set of substrates that they use. Enzymes are sensitive to temperature, and they can be sensitive to pH. For example, the effects of temperature. Enzymes have an optimal temperature at which they function most efficiently. In the case of an inorganic catalyst, for example, a metal helping catalyze a reaction, the reaction rate increases with temperature of the system. So you keep increasing the temperature of your system, the reaction rate keeps going up and up. So you can use very, very high temperature to accelerate your reaction. Enzymes aren't the same. They're normally exist as some sort of at body temperature. So you can think about the enzymes in our body. They would like to have an optimal temperature around an optimal reaction around body temperature. Or in vitro assays, you can look at enzyme activity. They may increase with temperature, but as you get too high in temperature, the enzyme activity declines due to denaturation of the enzyme. So, just for example, each enzyme has a maximum activity or particular temperature where they operate. So this one, it goes up above 25, and then once you get a little bit higher, it starts to go down as the enzyme denatures. So typically enzymes denature above 45 to 50 degrees C, and they have an enzyme optimal temperature of around 37 degrees C. This is true for most bacteria, most enzymes in humans. There are some exceptions for bacteria that grow in very high temperatures where their optimal temperature is much higher, and those have actually been exploited for biotechnology type of use. But typically your enzyme activity optimal temperature matches the physiological temperature that organism lives in. pH can also affect enzyme activity. So just like temperature, every enzyme has a pH optimal. This is due to the chemistry that's happening with ionizable amino acids like histidine, glutamate, cysteine. For example, these enzymes are typically at active sites impacting the reaction. So if you can imagine, if you're adding or subtracting a proton to one of those sites on the enzyme, you can dramatically impact the reaction rate. So typically cytosolic enzymes have a pH optimal of 7 to 8. That's typically the pH in the cytosol. Some examples where there's dramatically different optimal pH. For example, pepsin. This is secreted by gastric cells and functions in the gastric juice, which is very acidic. So this enzyme has an optimum of around 1.5 to 2. Whereas trypsin and chymotrypsin, they are in the pancreatic juice that has an alkaline pH, so they have a higher pH optimal. And lysosomal enzymes have an acidic pH optimum. Now, the pH sensitivity of enzyme results from the effect of the pH on the ionic charge of amino acid side chains of enzymes, just like I mentioned above. So various solutes, substrates, products, metal ions, and regulatory molecules can also affect the rate of enzyme reactions, and we'll talk about these as we move through the lecture. So here's an example for trypsin. You can see the activity on the y-axis as a function of pH on the x-axis. It has an optimal pH of around 7 or 8, whereas pepsin has an optimal pH around 2. And as I mentioned, most enzymes have an optimal pH around 7, but in different compartments in your body, you may have different pH optimum. Now let's talk a little bit about enzyme classification. There are 6 high-level classes of enzymes: oxoreductases, transferases, hydrolases, lyases, also known as synthases, isomerases, and ligases, also known as synthetases. Synthetases require ATP. And we'll talk through each of these and what they do. Before that, let's just talk a little bit about the naming conventions of enzymes. Typically, enzymes have an "ase" at the end of it. They're formed by adding this suffix "ase" to the name of the substrate. For example, tyrosinase catalyzes the oxidation of tyrosine. Cellulase catalyzes the hydrolysis of cellulose. But common names don't often describe the chemistry of the reaction. There's a lot of common enzymes like pepsin, catalase, trypsin, where the name itself really doesn't characterize exactly what it does. So, there are official naming conventions for enzymes, but a lot of times these aren't followed. So sometimes the name may mean something, and other times it may just be a common name. So as I mentioned, all enzymes are classified into the 6 categories according to the type of reaction they catalyze. And it has an official international name ending in this "ase," but a lot of the times they use these common names. There are also enzyme classification numbers. You may have seen enzymes like EC numbers. And basically the first digit refers to the class of the enzyme. So this would be a 1, 2, 3, 4, 5, or 6. The second to a subclass, and then subclass, and so on. So you can get very, very specific as you go down. When a new enzyme is discovered, they may know that it belongs to this class and subclass, but they don't know the exact substrate, so it would just be classified as 2.3, for example. But you can get very specific all the way down to the exact reaction that it catalyzes. Let's talk through the 6 classes of enzymes. The first one being oxioreductases. So this catalyzes an oxidation reduction reaction. For example, lactate dehydrogenase takes lactate in the reduced form and NAD plus in the oxidized form, and basically does an oxidation reduction where lactate gets oxidized to pyruvate and NAD gets reduced to NADH. Other enzymes in this class include oxidases, peroxidases, and dehydrogenases. The second group, transferases, catalyzes group transfer reactions. So alanine transaminase, for example, catalyzes the transfer of an amine group. So if you have L-alanine, the amine group is transferred from alpha ketoglutarate to end up with L-glutamate. The third class is hydrolases. It catalyzes hydrolysis reactions where water is the acceptor of the transfer group. A very simple example is pyrophosphatase, where if you have pyrophosphate and you add water with the enzyme, you get water hydrolysis across this bond to end up with 2 phosphate groups. The fourth enzyme classification are lyases. It catalyzes the lysis of a substrate, generating a double bond and a non-hydrolytic, non-oxidative elimination. One example is pyruvate decarboxylase. It takes pyruvate and a proton to form carbon dioxide and acetaldehyde. The fifth category is isomerases. This catalyzes the isomerization of a substrate. So for example, alanine racemase just takes L-alanine and turns it into D-alanine, basically swapping the direction of these 2 groups, flipping it as a mirror image essentially. The sixth one is ligases, otherwise known as synthetases. It catalyzes the ligation or joining of two substrates, but it requires a chemical energy like ATP. So an example here would be glutamine synthetase. It takes L-glutamate with ATP and turns it into L-glutamine using the energy hydrolysis of ATP to ADP in the process. And some of my later lectures will cover how this coupling reaction occurs. Those were the enzyme classification. Let's talk through a little bit about the structure of enzymes. So enzymes can be built of different parts. Sometimes enzymes are just the protein itself. In those cases, you can just think of it as a simple enzyme. It's just the protein. There are also other enzymes that are complex or holoenzymes, so it contains a protein part and a non-protein part. In this case, you'll see the protein part referred to as the apoenzyme because this is basically the enzyme without its cofactor. And then the cofactor would be the non-protein part. And there's different types of cofactors. There's prosthetic groups. Usually these are small inorganic molecules or atoms, and they're usually tightly bound to the enzyme. Cofactors can also be coenzyme, which is a different class where these are larger organic molecules that are loosely bound to the enzyme. Let's talk through the cofactors a little bit. First, the prosthetic groups. An example here would be metalloenzymes that contain firmly bound metal ions at the active site. For example, this carbonic anhydrase contains zinc at the active site. So this would be an example of a prosthetic group. On the other hand, coenzymes are larger in nature. They can act as group transfer reagents. So hydrogens, electrons, or groups of atoms can be transferred. The coenzymes can be classified, either subclassified even further into metabolite coenzymes. So these are synthesized from common metabolites, or vitamin-derived coenzymes. So obviously derived from vitamins. These vitamins can't be synthesized by mammals, so they must be obtained in the food we eat. An example of a coenzyme is a TPP, thiamine pyrophosphate. The structure shown here. And throughout my lectures, I'll show a lot of structures. There's not going to be structures that need to be drawn on the tests, but this just gives you a flavor of what some of these look like. But TPP is a derivative of vitamin B1. It participates in reactions of oxidative decarboxylation and transketolase enzyme reactions. It's just an example of a coenzyme. Another example is pyridoxal phosphate, or PLP. It's derived from vitamin B6. It's a coenzyme for enzymes catalyzing reactions involving amino acid metabolism like isomerizations, decarboxylations, transamidations. The enzyme active site itself is where all the action happens. So the substrate is usually a relatively small molecule, and the enzyme is a large protein molecule, obviously. So the substrate must bind to a very specific area on the enzyme, and this is what we call the active site. There are a few characteristics of active sites. The first one being specificity. It has very absolute or relative specificity in some cases. You may have a couple of different substrates that are very close in chemical nature that are specific for that active site. Enzymes active sites are also stereospecific, so they may only work on the L versus the D form of the substrate, for example. The active site itself is a small three-dimensional region of the protein. The substrate interacts with about 3 to 5 amino acid residues, and these residues can be far apart in linear sequence space. It binds the substrates through multiple weak interactions or non-covalent bonds, and there are contact and catalytic regions in the active site. Here's an example of the lysozyme active site. It consists of 6 amino acid residues, which are far apart in the linear sequence. So, for example, the active site residues span from residue 35 all the way up to 108, but 35 and 108 are very close in three-dimensional space, even though in linear space. And for the primary amino acid sequence, they're far apart. The active site contains functional groups like OH groups, NH groups, carboxylic acid groups. For example, here's an active site that has a threonine side chain that can perform hydrogen bonding with a substrate such as uracil. The serine side chain can form these hydrogen bonding networks. So you get these multiple weak interaction non-covalent bonds with the substrate. It can not only be hydrogen bonds, it can also be more hydrophobic interactions. The main theory, or the easiest way to think about active site substrate interactions, is the lock and key model. The enzyme active site being the lock, and it can accept only a very specific type of substrate, or the key. So the enzyme can have these different sites as part of the enzyme, and the substrate fits as a key into those different sites. So the substrate and enzyme come together to form the enzyme substrate complex. That's the main structure of enzymes. Let's talk through the properties of enzymes, and then we'll talk about the Michaelis-Menten equation. I touched on this briefly, but the specificity of enzymes can either be absolute or relative. In the case of absolute, one enzyme acts on only one substrate. And shown here are some examples. For example, arginase only can act on arginine. Relative enzymes is when one enzyme can act on different substrates that have the same type of bond. For example, pepsin is a protease that cleaves different sites in the protein, but they have similar, same bond types and similar type of substrate moieties. A third property is stereospecificity. So some enzymes can catalyze the transformation of only substrates which are in certain geometrical conformations, such as a cis or trans. This will come into play when we talk about lipid metabolism, some of the enzymes that work on cis versus trans lipids, cis versus trans double bonds in lipids. Let's take the catalytic site of these three different substrates. So for example, in the case of trypsin, three different enzymes that use three different substrates. In the case of trypsin, again, this is a protease that cleaves after on the C-terminal end of lysines and arginines. So in the case of trypsin, the active site looks like this, where you have your substrate, in this case a lysine, in the pocket, and you get a salt bridge from a carboxylic acid side chain here, and so that helps orient the substrate. Chymotrypsin performs the same type of chemistry, except for it cleaves the C-terminal end of a hydrophobic residue, such as phenylalanine. So the phenylalanine sits in this hydrophobic pocket in the case of the chymotrypsin enzyme. Elastase is cleaves after small aliphatic residues such as glycine, and you can see it has a small hydrophobic pocket. Even though they're all doing the same type of chemistry, cleaving at this bond here at the C-terminal end, they have different substrate specificities because of the pockets that are formed by the enzyme active site. As a bit of a side note, sometimes you'll see enzyme activity defined as in terms of international units. The international unit, one international unit of an enzyme catalyzes the conversion of 1 micromole of substrate to product per minute. So when you buy enzymes for research, or even when you buy cosmetic forms of enzymes, for example, botulinum toxin for cosmetics or for medicinal purposes, Botox, for example, you have a specific number of units, and that unit is defining the enzyme activity. The specific activity is the measure of international units per milligram of protein. So total activity is just the units, but then how specific it is, so it's X number of units per X milligrams of protein. These will come into play when we talk about Michaelis-Menten kinetics. So it's important to understand total activity versus specific activity. Okay, let's talk about Michaelis-Menten kinetics. Let's first think about enzymes, the enzyme reaction in time coordinate on the x-axis. So if we look at the concentration of the various things that are in the reaction, the enzyme itself stays relatively constant. So we're not losing any enzyme, we're not gaining any enzyme, it's just moving between the enzyme and enzyme substrate complex. So the free enzyme dips down a little bit in the middle while the enzyme substrate complex goes up, but in the end you end up with the same concentration of enzyme as you had in the beginning. Whereas the enzyme substrate, starts at 0, goes up, and comes back down to 0 eventually. In contrast, the substrate concentration, starts at its highest point, and then over time, you can deplete the complete substrate concentration, depending on the enzyme. Sometimes, depending on the equilibrium constant, you may not deplete the entire concentration of the substrate. But just giving a simple example here, in which all the substrate is consumed, and then over time you get all the product being formed. So this is the reaction coordinates in terms of time. If we think about this initial velocity, the initial velocity of that enzyme reaction, so just that initial jump in the curve from the previous slide, if we think about that in reaction in terms of substrate concentration, so as we add more and more substrate, you get a curve that looks like this. And this is a typical Michaelis-Menten curve, in which you add more and more substrate, and the velocity of reaction goes up and up and up until eventually you have enough substrate in there to where the reaction cannot go any faster. And the point at which this half of this max reaction occurs is your Michaelis constant, or your Km. So the rate of catalysis at a fixed enzyme concentration, or E, the initial velocity is almost linearly proportional to the substrate concentration, S, when S is small, but nearly independent of S when S is large. So when we're at the bottom of that curve on the previous slide, as you increase substrate concentration, the initial velocity increases. But then at the upper part of the curve, it levels off, so the rate of the reaction is independent of the substrate concentration. So as you can see in the previous slide, it goes up and then saturates at the top point. Let's talk through this a little bit in terms of reactions and how Leonor Michaelis and Maud Menten first came up with this. So they first understood this curve, explained this curve back in 1913. This reaction can be let's think about a very simple reaction in which you have an enzyme and one substrate where K1 and K-1 are rate constants, on-off constants to form the ES, or the enzyme substrate complex. And then catalysis occurs and you have release of the free enzyme and release of the product. So these are rate constants that indicate the speed of this reaction. But essentially you're going enzyme plus substrate to form the enzyme substrate complex, and then you get the reaction and release of the free enzyme again and the product. So the basic equation to describe this reaction that's derived by Michaelis and Menten is the V0, or initial velocity, at a particular substrate concentration S is equal to the Vmax, or the maximal velocity on that saturation curve, times the substrate concentration over the concentration of substrate plus Km, or this Michaelis constant. And again, the Michaelis constant is when the half is the substrate concentration at half maximal velocity. So this curve shows that. Again, this is just this simple enzyme plus substrate reaction. So if we think about titrating in substrate, the Km is when you have the substrate concentration at half Vmax. The Vmax is just the initial velocity, maximum initial velocity. And then V0, we're talking about, is just this initial velocity curve. So why do we care about this, and why is it important? Let me put this into context with an example. Here are two enzymes that catalyze the same reaction, glucose kinase and hexokinase. They both catalyze glucose plus ATP to form glucose 6-phosphate plus ADP. When we look at the Michaelis-Menten kinetics of both of these, they're drastically different. So hexokinase has a Km, a small Km of about 0.2 millimolar, the Vmax of about 0.1 millimolar. In contrast, glucose kinase has a much larger Vmax of about 1.5 micromolar. This is, sorry, written wrong. This should be millimolar. So it has a much higher Vmax, and the Km is much higher as well, about 10 millimolar. The reason for this is even though they are isozymes of each other, they're needed for different reasons. So hexokinase is found in all cells. It catalyzes the first step in glucose metabolism. Again, here's the reaction. It has a low Km for glucose and is inhibited by glucose 6-phosphate, its end product. The normal levels of glucose in blood are about 5 millimolar, and basically hexokinase efficiently catalyzes the reaction under these normal conditions. Glucose kinase is more sensitive to substrate concentration. So it's found in hepatocytes, which we'll talk about those in some later lectures. They store glucose as glycogen. It's also found in pancreatic cells. It has a much higher Km for glucose, and it's not inhibited by the product glucose 6-phosphate. So as you eat more and more glucose from a meal, it phosphorylates with increasing efficiency, so the reaction rate becomes higher and higher. So it needs to be more sensitive to glucose concentration. Just by way of showing a couple other example plots, so the plots I've been showing you, there's some other representations of Km curves. So typically you see it in terms of substrate on the x-axis and V0 on the y-axis. You can also do the reciprocal plot of that, called a Lineweaver-Burke plot, in which you get a linear plot. Essentially, it's just taking the reciprocal of each of these. And similarly, you can get an Eadie-Hofstede plot, which is also a linear plot. but they're all the same Km curves. It's just derivatives of each other. We've talked about the effect of substrate concentration on the reaction rate. What about the effect of enzyme concentration? It's much simpler. So as you increase enzyme concentration, your velocity keeps going up and up and up. So as long as you have enough substrate present, if you keep adding more and more enzyme, your velocity goes up. So that's Michaelis-Menten kinetics. What about if you have an inhibitor in the reaction that can slow that reaction down? Let's talk through the major types of enzyme inhibition. These are important because small molecule drugs are enzyme inhibitors, antibiotics are enzyme inhibitors. So the whole pharmaceutical industry, a lot of the pharmaceutical industry is based around inhibiting inhibiting enzymes with small molecules. Let's try to understand the mechanisms of enzyme inhibition. So in tissues and cell, different chemical agents can inhibit enzyme activity. You can also have endogenous chemicals like drugs inhibit those enzyme activities. The inhibitor binds to an enzyme and prevents the formation of the ES complex or breaking it down to the enzyme plus product. So essentially you're in that reaction coordinate, you're inhibiting it somewhere along the way. And depending on at which point you're inhibiting it, you're classified as a different type of enzyme inhibitor. Let's first talk about reversible inhibitors. So reversible inhibitors are after combining with the enzyme, you form an EI complex. It can rapidly dissociate. So the enzyme is inactive only when bound to the inhibitor. This EI complex is a non-covalent complex. It's held together by weak non-covalent interactions. And there are three basic types that we're going to talk about: competitive, uncompetitive, and non-competitive. First, competitive inhibition. In the case of a competitive inhibitor, the inhibitor has a structure similar to the substrate and can thus bind to the same active site. So essentially the enzyme can't differentiate between the substrate and the inhibitor. So when the inhibitor is bound to the enzyme, it prevents the substrate from binding. Then the inhibitor can be released by putting more and more substrate into the system. So with the lock and key type mechanism, the substrate binds to this pocket, the active site pocket on the enzyme. Competitive inhibitor binds to the same place. So, for example, an example of competitive inhibition is benzimidine, which competes for arginine for binding to trypsin. So trypsin, the native substrate of, arginine is part of a native substrate for trypsin. Benzimidine binds in that same pocket. What about non-competitive inhibition? Non-competitive inhibition is different in that the inhibitor binds to an enzyme site that's different from the active site. So it can bind to this, it can either bind to the enzyme or bind to the enzyme substrate complex. And so the inhibitor and the substrate can bind to the enzyme at the same time because it's at a different site. In this case, you can't overcome the inhibition just by adding more and more substrate because obviously it's at a different site on the enzyme. Uncompetitive inhibition is even different yet, in that uncompetitive inhibitors bind to the enzyme substrate complex, not to the free enzyme. So in the case where you have the free enzyme, you add the substrate, then you have the enzyme substrate complex. And the difference between uncompetitive inhibitor and non-competitive is uncompetitive can only bind to the enzyme substrate complex. So it needs the substrate to be there as part of the enzyme. So you usually see this in multi-substrate reactions. And again, you can't, like non-competitive inhibition, you can't overcome this just by adding more substrate because the non-competitive inhibitor is at a different site on the enzyme. To summarize these, you can see this chart here. So a competitive inhibitor binds to the same site on the enzyme. So it binds to the free enzyme shown here. Non-competitive inhibitor can bind to either the enzyme or the enzyme substrate complex. The uncompetitive inhibitor can only bind to the enzyme substrate complex. Here's what the Michaelis-Menten curves look like for all these. So if we have our normal Michaelis-Menten curve without any inhibitor, you can see here the Km is half the velocity for the normal enzyme. If we add a competitive inhibitor, it essentially shifts the curve to the right, but as you add more and more substrate, you can overcome that and you end up with the same Vmax. Non-competitive inhibitor, you have the same Km, but a decreased Vmax, and you can't overcome the velocity. You can't max out the Vmax to the normal enzyme levels just by adding more substrate. And the same story holds true with the uncompetitive inhibitor. The Km is increased and the Vmax is decreased. Those were reversible enzyme inhibitors. You can also have irreversible enzyme inhibition. In this case, you have very slow dissociation of the EI complex. So the enzyme inhibitor is tightly bound through a covalent interaction or non-covalent interaction if it's tight enough. So basically, you don't have dissociation of that complex, or it happens at a very, very slow rate. Some examples include group-specific reagents, substrate analogs, and suicide inhibitors. A good example of an irreversible inhibitor are antibiotics such as penicillin or cephalosporins. These are antibiotics, obviously, that are used, and they have very irreversible mechanisms, and they make very good antibiotics. An example of a group-specific reagent would be DIPF, which binds on acetylcholine esterase in our bodies. It can form a covalent bond with the hydroxyl group on serine, and in the process release a fluoride molecule. But it essentially inactivates this enzyme by covalently bonding to the serine in the active site. Example of a substrate analog that is structurally similar to the substrate that can covalently modify active residues is shown here. So the natural substrate for chymotrypsin is shown here. In that case, if you add TPCK, it reacts with the active site histidine, forming a covalent modification and inactivating the enzyme. Suicide inhibitors are special cases of irreversible inhibitors in which the inhibitor binds as a substrate and initially processed by the normal catalytic mechanism, but then generates a chemically reactive intermediate that inactivates the enzyme through covalent modification. So it's called suicide because the enzyme participates in its own irreversible inhibition. That's inhibition, which is one form of a regulatory mechanism. Let's pull this up a level and talk a little bit about regulatory mechanisms in general, either from an inhibition or an activation standpoint. So beyond just inhibition, there are other ways to regulate enzyme activity. You can do allosteric control, reversible covalent modifications, which we talked a little bit about. You can have isozymes, so enzymes that, like hexokinase that we talked about, that can catalyze the same reaction as other enzymes. They're just different forms, and they have different enzymatic properties. You can also have proteolytic activation, and we'll talk through some examples of each of these. First, allosteric enzymes. So allosteric enzymes have a second regulatory site called the allosteric site that is distinct from the active site. Allosteric enzymes contain more than one polypeptide chain, so they have a quaternary structure. So when we talked about quaternary structure in the previous lectures, allostery can come into play here with some of these enzymes. So it's not just that an allosteric site is on one part of a monomer, for example, it would be on a different quaternary site on the polypeptide, and a different subunit, I should say. So allosteric modulators bind non-covalently to this allosteric site, and they can regulate the enzyme activity via conformational changes. There are two types of modulators. It can either be an inhibitor or an activator. So the negative modulator or the inhibitor, it binds to the allosteric site and inhibits the action of the enzyme. Usually that inhibitor is the end product of the biosynthetic pathway to provide this end product or feedback inhibition. You can also have a positive modulator or an activator. It can bind to the allosteric site and stimulate activity. This is usually the substrate of the reaction to push forward the biosynthetic pathway. Here's an example or picture of inhibition versus activation. So in the case of inhibition, the inhibitor binds to the site such that the substrate can't bind as easily, whereas in activation, the activator binds to the allosteric site and opens up that pocket and provides the ability of the enzyme to bind the substrate better to push the reaction forward. So here's an example of the Km curve for what these would look like. An allosteric activator would push that Km down, whereas the inhibitor would push the Km forward, basically slowing down the reaction. Here's some examples of regulation of enzyme activity by covalent modification. So in this case, covalent attachment of a molecule to amino acid side chain of the protein can modify the activity of the enzyme. For example, phosphorylation. In the case of glycogen phosphorylase, you can get acetylation on histones, can modify activity. You can add fatty acid chains for signal transduction type of functions, ADP ribosylations, ubiquitization. So there's all types of these covalent modifications that occur to regulate enzymes throughout our body. That's allosteric regulation. Let's talk about isozymes. We talked about hexokinase early on in the lecture. So some metabolic processes are regulated by enzymes that exist in different forms, or isozymes. So they have the same reaction, but they have different Michaelis-Menten properties. So the actual amino acid sequence is different, but they catalyze the same reaction. Again, they can differ in kinetics, regulatory properties. They can have different coenzymes or different forms of coenzymes, and they can be distributed in cells differently. So you might have more of one isozyme in certain parts of the body and less in other parts of the body. They are actually encoded by different genes. An example would be lactate dehydrogenase. So this is lactate dehydrogenase is a tetramer. It's composed of two types of polypeptide chains, M and H, and the different isozymes have different numbers of these polypeptide chains. So for example, in the heart lysozyme has four of the H polypeptide chains, and the liver and the muscle is made up of M4. So for the H4 has the highest affinity for substrate, and it's best in aerobic environment versus in the muscle, and the liver it's best in the anaerobic environment. So these are useful for our body to metabolize lactate in different tissues in our body. It can also be exploited for diagnostic purposes, so you can use lactate dehydrogenase as well as other enzymes that have isozymes for diagnosis. Here's an example where you can look at the different forms of lactate dehydrogenase in serum using these different chromatograms. In the case where you're having an acute heart attack, you can have more of certain forms, and in other diseases you can have more of other types of forms of these isozymes. And so you can use this panel as a diagnostic to diagnose these different conditions. Here's some other examples where these enzymes are used for clinical diagnosis. So aspartate aminotransferase, for example, can be used to diagnose liver disease. Alanine aminotransferase can also be used to diagnose liver disease. Amylase can be, and if you look at amylase in our pancreas or even in our saliva, you can help diagnose acute pancreatitis or obstruction, biliary obstruction, for example. So I don't need to go through all these, but these are some examples where you can use these enzymes, in some case isozymes, for diagnosing different conditions. Just to touch on a few other mechanisms of regulation. Activation by proteolytic cleavage. So some enzymes are synthesized as inactive precursors. So they'll have a portion of their protein that causes it to be inactive. And you'll see them called zymogens in some case. And then what happens is you get part of that protein cleaved, a peptide gets cleaved off and allows activation of that enzyme. This is a one-shot deal, so these proteins are synthesized as inactive precursors. A peptide portion gets chopped off, and then the enzyme becomes activated. So why would we ever do this? Well, for some of these digestive enzymes, when they're made, translated by the ribosome, you wouldn't want an active protease or active digestive enzyme chewing everything out as soon as it's made. So it's synthesized as this inactive form, and then once it reaches the point in the body where it needs to be used, then it gets cleaved and activated. So digestive enzymes, for example, are synthesized this way. Blood clotting enzymes are synthesized this way. This is hugely important. So the blood cascade, coagulation cascade, you'll see a lot of this occurring. Otherwise, you'd have clotting in your blood going on all the time. Protein hormones is another example. Insulin is actually a protein that's first made as a zymogen, and then a peptide gets cleaved to make it active. Here's an example for trypsin. So trypsin is a protease that's first made as trypsinogen, so it's in active form. There's an enzyme that acts on it to activate trypsin, and then trypsin can act on other enzymes to activate them. So, example, elastase becomes activated by trypsin, chymotrypsin becomes activated by trypsin, and so on. Additionally, you can get regulation by multi-enzyme complexes. So different enzymes that catalyze sequential reactions can be bound together in some cases. When we talk through lipid biosynthesis, you'll see how this comes into play. And you can have different activities that can be found on so that's multi-enzyme complexes. When you talk about a multifunctional enzyme, you can actually have different activities on the same single multifunctional polypeptide. You can also get what's called metabolic channeling. So it's channeling of reactants between active sites. So this occurs when the product of one reaction is transferred directly to the active site without entering the bulk solvent. So it's basically you're just pumping that reaction through multiple enzymes. So you can imagine this can greatly increase the rate of reactions. So when we think about multi-enzyme complexes and multifunctional enzymes, these things are possible. And again, when we talk about lipid biosynthesis, this will come into play. That's it for this lecture. I'm going to leave you guys with this summary. Again, here are the main points that we we hit today, and I thank you for your time, and I'll see you next time.